Hi everyone, this video is about C3, C4, and CAM plants. So we'll talk about the concepts of transpiration and photorespiration and the different ways that the plants try to prevent it from happening. So first, let's talk about C3 plants. It lives in temperate conditions and it's the most common type of plant, so it's probably most of the plants that you would encounter. So for example, we have maple trees, wheat, and rice. The C3 plant does normal photosynthesis, and in this video I'm assuming you already have a background understanding of how photosynthesis works. So the C3 plant follows that process that you've probably learned about in the textbook, and later we'll talk about how C4 and CAM plants have adapted their photosynthesis to be different. However, the C3 plant has a problem of photorespiration when it does photosynthesis. So let's go into detail to see how that happens. This is a cross-section diagram of a C3 plant leaf. So first I would like to note that this is very much simplified. So the actual plant leaf would have many cells within it, and photosynthesis would specifically be taking place in the mesophyll cells. And also these molecules are much too large for the proportion here, but I've drawn attention to oxygen, carbon dioxide, and water because those are involved in photosynthesis. And finally, I would like to note that there's stomata, which are holes at the bottom of the leaf, and the plant is able to open and close the stomata. So during photosynthesis, the plant is using up CO2 and it's making a waste product of oxygen. And so these stomata allow CO2 to diffuse into the leaf and the oxygen can exit so that there's not too much or too little of each substance. So you can see that there's CO2 that's outside in the air that's going into the plant leaf while the oxygen is exiting. And now let's discuss transpiration, which is a problem that can occur due to the stomata being open. If it's really hot outside, the water can actually exit the leaf by transpiration, which basically just means that it evaporates. And so as a result of transpiration, the plant can become dehydrated because of water loss. And since water is necessary for photosynthesis, it's not sustainable for the plant to have no water for long periods of time. So there is a solution that the plant can do to help fix the problem of transpiration. If it closes up its stomata, then the water cannot exit. But this leads to a different problem, which is called photorespiration. Because the stomata are closed, this also prevents the gas exchange of CO2 and oxygen. So what's going to happen is a buildup of oxygen and a decrease in the amount of CO2. Remember that oxygen is a waste product of photosynthesis. So as the plant does photosynthesis, it's making more and more oxygen, which now has no way of escaping the leaf. And since the plant needs CO2 as a reactant in photosynthesis, it's using up the CO2 without any new CO2 coming in to replace it. So this actually leads to a problem. So over here we have the rubisco enzyme, whose role in the Calvin cycle is to join carbon dioxide to this compound called RUBP. And this will ultimately make G3P, which can be made into glucose and other sugars as food for the plant. However, rubisco is not very picky, so it's actually able to join oxygen to the RUBP rather than joining carbon dioxide, and that is called photorespiration. So when there is such a low concentration of carbon dioxide and such a high concentration of oxygen due to the stomata being closed, it becomes more likely that rubisco is going to join oxygen rather than carbon dioxide, and this is also more likely to happen at higher temperatures. So now what we have is what I'm going to call a useless product. So why is it a problem that we're making this? Well, first of all, the Calvin cycle is using up ATP in the process of making this. So we've basically wasted some of the ATP making something that we don't even want. And also, although part of this product can be made into G3P, which is what we want, it only makes one G3P rather than the usual two G3Ps that you would make if we had joined carbon dioxide here instead of oxygen. So we're not making what we're trying to make. And the rest of this compound ends up being disposed of, which requires energy. And finally, when the piece is disposed of, it removes some of the carbon from the Calvin cycle. So we're going to have to fix even more carbon dioxide to make up for that lost carbon. So as you can see, photorespiration is not good, but C4 and CAM plants actually solve the problem by adapting their method of photosynthesis. Let's discuss the C4 plants first. So it lives in hot, dry climates where the plants have to close their stomata a lot due to the hot weather to try to conserve water. So it's worth it for C4 plants to try to stop photorespiration. So examples are corn, sugarcane, and some types of grasses. So for example, ornamental grass. And it uses the C4 pathway to reduce photorespiration. So let's take a look at how that works. This is a C4 plant leaf cross-section, so it looks different than C3. C3 would have the mesophyll cells just kind of spread out in the leaf, and the gases can go directly to and from the mesophyll cells for photosynthesis to occur. But in C4, the mesophyll cells surround these other type of cells called bundle sheath cells. And so the light-dependent part of photosynthesis happens outside in the mesophyll cells, just as it would happen in the C3 plant, but the Calvin cycle actually takes place in the bundle sheath cell instead. Let's zoom in. So this is the mesophyll cell, and this is the bundle sheath cell. So remember that the mesophyll cell is on the outside layer, while the bundle sheath cell is on the inside layer. And so most of the oxygen and carbon dioxide are going to be concentrated out here, because the bundle sheath cell is further away, so they would be less likely to diffuse all the way in here. They're mostly going to be outside here. And inside the mesophyll cell, we have an enzyme that's called PEP carboxylase, and we also have a compound called PEP. So PEP carboxylase will actually join CO2 and PEP together, and it's very selective. It will only choose CO2, it will not choose oxygen, even though there's such a high concentration of oxygen outside. Then this is converted into a compound that eventually becomes malate, and it moves over to the bundle sheath cell, where it deposits the carbon dioxide. And in the bundle sheath cell, we have Rubisco and the rest of the Calvin cycle, so now the CO2 is available for the Rubisco to use. And so because of the way that the mesophyll cell is kind of acting like a shuttle system for the carbon dioxide, there's going to be a really high concentration of carbon dioxide near the Rubisco, so it's unlikely that photorespiration will occur. So this is the C4 solution to photorespiration. However, it requires extra energy to have all these extra steps within the mesophyll cell, but it's worth it in this case because the plant lives in such a hot environment. So the stomata have to be closed a lot, and therefore photorespiration would become a real problem if we didn't have the C4 pathway. And finally, we have cam plants. So it lives in an even hotter and drier climate than the C4 plant. So this is a type of plant you might find in the desert, for example, cactus or succulents. And it uses the CAM pathway to reduce photorespiration. So let's go in detail of how this works. The CAM plant leaf structure looks more similar to C3 because the photosynthesis all takes place in the mesophyll cells, and it doesn't have that C4 structure with the two layers of cells. So how it works is at nighttime, the stomata are open and it allows the gas exchange, so oxygen can exit and CO2 can enter. And this is pretty rare because most plants would have the stomata closed during the night to conserve water, because there's no reason to have gas exchange at the nighttime when we're not actually doing photosynthesis. But for the cam plants, they actually have the stomata open at nighttime and always closed during the day. So the gas exchange will occur during the nighttime where CO2 can diffuse inside of the leaf. Now we perform that same C4 pathway that we just talked about, where the PEP carboxylase enzyme will join the CO2 to the PEP, and it goes through some further reactions to eventually become malate or a similar compound. And this compound is stored up in the cell vacuoles, because remember that the light-dependent part of photosynthesis cannot be performed during the night, so the carbon dioxide is not needed right away. So in this animation, I am showing that the carbon dioxide is being stored up. Then in the daytime, the cam plant has its stomata always closed, but at this point the CO2 is released again, and so the Calvin cycle can be performed. And there's a high enough level of carbon dioxide that it's unlikely that photorespiration will occur. So this is how it solves the problem of photorespiration. And also the cam plant is really good at conserving water because it only opens the stomata at night, where it's usually a cooler temperature, so there would be less transpiration. But as I said before, it takes energy to perform these extra steps of fixing the CO2 twice, so it's kind of a trade-off about which method that the plant uses. C3 plants live in a cool and wet enough environment that photorespiration isn't really enough of a problem to require that extra energy use of fixing the CO2 twice. However, the C4 and the cam plants live in hotter environments where they have to close the stomata a lot more to prevent water loss, and therefore photorespiration could be a much bigger problem for them if they didn't use those alternate pathways. And since it uses a lot of energy to deal with the byproducts of photorespiration, it's actually better for them to use a little bit of energy in fixing CO2 twice, rather than losing so much more energy by having photorespiration happen. So that's all for this video, and I hope this helps you understand the difference between C3, C4, and cam plants. Thank you for watching.